Mic check, check, check. Mic check, check. Mic check. Siapa?
நீங்கம்மா
வெளியே போ. ஆலன் எதுக்கு விற்கிறதில்ல உங்களுக்கு பைத்தியக்கார கம்பெனி ஆலனே?
చికెన్ అంటే ఎదురున్నదే అన్నకి నా వెళ్ళుదా
Very good evening, children. Can you hear me? Are you able to see me? Can you hear me, children? Am I audible to you all? Okay, children, and today's class is again the continuation topic, the same topic, the human health and diseases. Last class, I hope you remember that I've explained you about the various lymphoid organs, and today's class is about antigens and its types, and then I'm going to explain to you about the structure of antibody and about vaccines, types of vaccines, and immune response. These are the topics what we are going to cover today. First, let me explain you about the term antigen and antibodies. If suppose any pathogen enters inside our body, we know what is pathogen, right? The disease causing organism. When any disease causing organism enters into our body, it releases a proteinaceous substance called antigen. That pathogen that enters into the body, it releases a substance called antigen. Those antigen, it get incorporated in various metabolic activities that takes place in the body, and it causes diseases. This is how diseases are caused. Now let me explain about what is an antigen and how a body responds to that. Antigen, it is a proteinaceous substance. Sometimes it is a sugar molecule like polysaccharides. When the antigens are released from the pathogen, our body start responding to that. The immune system, it start responding to that. With the help of T cells, it start producing the specific antibodies, or sometimes it shows the response with the help of B cells. These antigens that are released, they contain proteins, lipids, sometimes they have carbohydrates or even nucleic acids. If you see the diagram here, this is a pathogen, and pathogen releases antigen. According to the antigen, the antibodies are produced in the body. According to the antigen, the structure of the antigen, the antibodies are produced. Maybe you could have asked the question about the viruses. Why there is no antibody for virus? Because viruses, it does not have a definite shape, and viruses are pleomorphic organisms. What the virus it will do, it keep on changing its structure. There are a lot of morphological changes takes place in the virus. That is one of the reason why virus, there is no specific antibody for viral diseases. So each and every antigen that is released by the pathogen, it has a specific feature or a region that is called epitope. And similarly, the antigen, they have the epitope. Similarly, according to the epitope, the antibodies, they have a structure, a region called paratope. Always the epitope, it matches with the paratope. Epitopes are present in the antigen, whereas the paratopes are present in the antibody. So here you can see that this is called epitope, this is called paratope. And when there is an antigen, our body, according to the epitope, the antibodies are produced, or the paratope, it is modified in such a way that it attacks the antigen properly. When these antigens are there in the body, it stimulate our immune system to produce immunogens. What are immunogens? The molecules that stimulate immune response. That is called as immunogens. When antigens are there, it starts reaction, thereby immunogens are produced. What are immunogens? Molecules that stimulate the immune response. They are called as immunogens. And epitope, I have explained the epitope. The antigen, it has a specific region where the antibody binds to that. That is called epitope. Paratope. Paratope, it is the region that is present in the antibody which attaches to the antigen or epitope. Always the paratope, it binds with the epitope. Thereby it brings the immune response. The epitope, it is an antigenic determinant, and that is the active part of the antigens. Similarly, paratope, it is the binding site of the antibody. So according to the epitope, the paratope, it will be there. And this epitope, it has active regions of immunogens. Active regions of immunogens. So they can activate the immune system and bring about antigenic responses. So that epitope, it is also called as antigenic determinants. Antigenic determinants. Next, we are going to talk about the types of antigens. Types of antigen. What are the types of antigens? There are antigens which are exogenous, from outside. There are antigens from inside, they are called endogenous antigens. There are antigens which are called autoantigens, alloantigens and heterophile antigens. These are the 5 different types of antigens are there. I am going to explain in detail about all these types of antigens. Exo means outer. If the antigens are from outside the organism or outside cell, which enters into a body and which causes responses. For example, the microorganisms, the virus, bacteria, fungi, the protozoans, pollen grains, dust particles, pollutants. If there is a dust, if you inhale the dust, immediately we will sneeze that. That is a response. That is because of exogenous substances. There are antigens that are formed within our body. They are called as endogenous antigen and also called as self-antigen. For example, if you take the RBC, the surface of the RBC, it contains antigens that help us to identify the blood group. Those antigens are called as endogenous antigens. For these endogenous antigens, our body immune system will not show any response to that. Mostly the response, it will be for exogenous antigens. Autoantigens, auto means self antigens. Sometimes the person own self antigens are there. Example, thyroglobulin, DNA, corneal tissue, these are called autoantigens, self antigens. Alloantigens, antigens that are found in different members of the same species. For example, the RBC antigens, A, B, it is there. These antigens are found in different members of the same species. A blood group between human beings, they are all same. That's called alloantigens. Heterophile antigens, identical antigens found in the cells of different species. If the antigens are found in the same species, it is called alloantigens. If the antigens are present in different species, then it is called as heterophile antigens. Cross-reacting microbial antigens, Forestman antigens, these are examples for that. These are the 5 different types of antigens. So according to the antigen, the immune system shows responses. Sometimes when we inhale something, immediately we will sneeze that to send that microorganism or dust particles out. That is also a response. Next we are going to talk about antibodies. As I mentioned earlier, according to the antigens, antibodies are formed in a body. According to the antigens, antibodies are formed. These antibodies that are formed, it is mainly according to the antigens. Antibodies are mainly again a proteinaceous substance, a protein molecule. They are mainly synthesized after antigenic stimulation. The antigen, the pathogen enters inside the body, it produces antigen. According to the antigen, the antibodies are formed by the immune systems. When the pathogen enters inside the body, first the B lymphocytes produce an protein called antibodies to fight with them. These are secreted in response to the antigen by the effect of B cells called plasma cells. These plasma cells are also involved in production of the antibodies. This is a typical antibody structure, and these antibodies are classified into 5 types based on their physiological and biochemical properties. There are 5 different types of immunoglobulins are there. These antibodies are immunoglobulins, proteins. There are 5 different immunoglobulins are there. That's what IG stands for immunoglobulin. We can see IgG, that is called gamma, IgM mu, IgA alpha, IgD delta, and IgE epsilon. This is a typical structure of an antibody. These antibodies, they have, usually it will be Y-shaped structure. You can see it here, Y-shaped structure, which has 4 polypeptides. In that, 2 identical light chain, 2 identical heavy chain. The smaller one, which is what you are seeing it here, this is called light chain. This is called 2 heavy chains. So 2 identical light chains it will be there. The molecular weight is 25,000 Daltons. Approximately, it consists of 214 different types of amino acids, because the amino acids are the one which forms the protein. Since these are all proteins, the short light chain itself, it has 214 amino acids. Similarly, the heavy chain, a pair of heavy chain is there. This heavy chain, it contains 50,000 Daltons, and approximately it contains 450 amino acids. And this light chain and heavy chain, they are joined together by disulfide bond. Can you see it here? This is the disulfide bond. The disulfide bond, it helps the chain. That is the role of the disulfide bonds. Next is the heavy chain. This heavy chain has a flexible hinge junction. This is called hinge junction in the middle. This is called hinge junctions. And each L chain and H chain. L chain stands for light chain, H for heavy chain. It has 2 terminals. They have N terminal and C terminal. The N terminal, amino terminals. The C terminal is called carboxyl terminals. Acidic terminal, it will be there. Can you see it here? This is called COH. That's called carboxyl or C terminals. And apart from that, each chain has 2 regions are there. Can you see it here? This is called the variable regions. This is called constant regions. Variable region at one end and constant region at another end. So all these are all the variable regions. These are all the variable regions. These are all the constant regions. Mostly the COH end, it is a constant region, whereas the amino region, that is called the N terminal region, it has the variable regions. So what is the variable region? The variation, it is there in the amino acid. That's why it is called variable regions. Now I am going to explain about the variable regions. As I mentioned earlier, the variable regions are mostly present in the N-terminal portion of the molecules. Why it is variable region? Because it contains different types of amino acids are there. The amino acid sequence are different. And this high variability, what you can see it in the variable region, H region and L region, they are called as hot spots or hyper variable regions. They have lot of hot spots are there. The hot spots are the region for antigen binding site. That means the paratopes are present in the hot spots or in the variable regions. We know what is paratope. Paratope is a structure that attaches to the antigen. That's why the variable regions, they have antigen binding site. They have antigen binding site, it will be there. Next is the constant region. The constant region, this, all these are all the constant region. This is called fragment of constant region, FC region. They don't vary in amino acid sequence. The amino acid sequence are similar, whereas in the variable regions, the amino acid sequence, it will vary. That is the difference between the variable region and constant regions. Next, we are going to talk about the functions of immunoglobulin. Before that, I want you to draw this diagram and mark this also, the NH2, the amino region, COH region. I'll give you 5 minutes time. Just copy the diagram and draw the diagram and post it to in the WhatsApp group to your respective teachers. Madesh, you can send it to me. Draw the diagram. I'll give you 5 minutes time. Do it fast. Children, have you finished? Children, have you done, children? We move on to the next topic. The next topic is about the functions of immunoglobulin. The functions of immunoglobulin, there are 4 different ways by which it can bring down the antigenic responses. One is agglutination, next one is precipitation, opsonization and neutralization. These are the 4 different modes by which it will does that. First one I am going to explain to you about agglutination process. Agglutination. Whenever a particulate antigen, what is particulate antigen? It is a bacteria or a pathogen or any antigen. When it interacts with the antibody, these antibodies, it joins them. It forms agglutination, clumping together, joining all of them together. That is called agglutination process. The antibody brings that agglutination, so they are called as agglutinin. So they can agglutinate, they clump all the antibodies, clump all the pathogen or antigen together. That is called agglutination process. Next is called precipitation process. Precipitation. What is precipitation? In the precipitation process, it is a reaction between a soluble antigen and antibody. It can bring about precipitation, like curd formation. It will be like that. The antibodies, it bring about precipitate formation and reacting with the antigen. So that is called, those antibodies are called as precipitants. They can clump them together, they can form a precipitate. Next is called neutralization process. In neutralization reaction, the antigen antibody reaction takes place where it will interact and elimination of harmful effects of the pathogen, it will be done. That's called neutralize. The antigens, it can cause lot of disturbances. So these antibody, it neutralize that. So antigens, they release lot of toxins, poisons. Antibodies, it act against that and neutralize that. So they are called as antitoxins. Antigen produces toxin. These antibodies, it neutralize that. So they are called as antitoxins. And these antibodies are highly specific. It is according to the particular antigen. It will activate the antigens, it will activate the stimulation process where antibodies, it neutralize that. It bring down the effect of it. They are highly specific for a particular disease, a particular antibody. Next is opsonization process. In this process, a pathogen, it will be marked. It will be identified and marked of ingestion. And then what will happen? Digestion process takes place. In opsonization process, the pathogen, it will be marked. Think that this is a bacteria. The bacteria, it will be marked first. You can see that this is the bacteria. The bacteria is now, it is marked. Now the binding of opsin, that is antibody, takes place. So this is a pathogen. This is a pathogen. The pathogen is now surrounded by the antibody. That is called opsin. This process is called opsonization process. And this antibody, it binds to a receptor of a pathogen cell membrane. So this is the outer membrane of the pathogen. It gets attaches to that, and then phagocytosis process takes place. What is phagocytosis? Bringing down inside the macrophage. Macrophages are cells that engulf the pathogen and kills them. When they take the pathogen inside, with the help of the lysosome, it will digest them. When the pathogen is brought inside, it forms a phagosome. With that phagosome, the lysosomes attaches. Phagosome is formed. The phagosomes contains what? The bacteria. Now, the lysosome, it attaches to that. Now, phagosome, lysosome, it join together and it forms phagolysosomes. So lysosomes contains digestive enzymes, hydrolytic enzymes. With the help of those enzymes, it kills the pathogens. That process is called as opsonization process. Here the pathogens are coated with a substance called as opsonin. With the help of that opsonin that is done by the antibodies, they will be easily taken up by the phagocytic cells. This process is called opsonization process. Next we are going to talk about vaccines. Before this, children, if you have any doubt, let me know it. If you have any doubt, let me know it. Otherwise, we'll move on to the next topic. Children, do you have any doubt? Okay, the next topic is about vaccines. We know what are vaccines. Vaccines are all substances that are introduced into a body to bring immune response. These vaccines are, it is given to a body in two forms. It will be there in the form of injections, or through orally they will give the vaccines. These vaccines are substances that are produced from the microorganisms, or even sometimes a weakened disease causing pathogen, it will be given to a body, or a dead pathogen or a portion of the pathogen, it will be introduced into a body. When something is introduced into a body, our body, it starts reacting to that. When our body, when they inject the dead pathogen into the body, just let us assume that they are injecting some weakened or dead pathogen into the body. Our immune system will not know whether it's dead or alive, or weakened pathogens. But this weakened pathogen, it will not cause any disease to our body, but they have the antigens. they have the antigens. When they have the antigens, our body start producing antibody against that. After a quite some time, our body will have antibodies. It will produce antibodies according to the vaccine given. The vaccine for common cold is available. The vaccine for flus are available. The vaccine for tetanus. Like that, there are different vaccines are available now. Now we have even adult vaccines are available. Very soon we are going to get the COVID vaccines. This vaccination, it induces our body to start producing antibodies. It will stimulate our body to produce the antibodies by the host cells. But it will not cause any disease, only it can stimulate, the antigenic stimulation it will be there in the body. This vaccination was first initiated by Edward Jenner in the year 1790. He observed that the milkmaids did not contract smallpox, the smallpox virus. They don't get smallpox virus because they are exposed to similar but milder form of diseases called cowpox. Because these milkmaids, they keep on working with the cow, they'll get that cowpox. So when they have that cowpox, which is milder to the human beings, they will not get the smallpox. So he identified that. So previously, if you have a milder infection, that help us to recover the virulent infections. So he has identified the vaccines there. So Edward Jenner, he infected the milder pathogen to a 8-year-old boy. It's called James Phillips. This 8-year-old boy was injected with a cowpox. We know what will happen if cowpox is a milder viral diseases. The same boy, after 2 months, he was infected with smallpox. Normally, when a person is first infected with smallpox, he'll get the disease. But this particular child, who is 8 years old, it did not get the smallpox. He did not suffer with the smallpox because he's already exposed to the cowpox, and the body start producing antibody for that. The antibody that are formed earlier due to cowpox, it protected the child from smallpox diseases. That is the reason why the term vaccine, it means cow. Vaka means cow, because he has taken the word cow there. That's why it is given as vaccine. Vaccine usually develop acquired immunity. What is acquired immunity? Immunity that will develop after birth. In that we have active and passive. Active means you will be exposed to a disease and will develop antibody. That is called active. Passive means by taking vaccines. And also Pasteur confirmed the Edward Jenner's finding, and he's produced lot of vaccines for anthrax, rabies and cholera. Now I am going to explain to about the classification of the types of vaccines or classification of vaccines. There are different kinds of vaccines are there. We have first generation vaccine, second generation vaccine and third generation vaccines. I am going to explain about the generations of vaccines. First generation vaccine. In that vaccine, they will give attenuated pathogen. That means weakened pathogen. The same pathogen that causes a particular disease, it will be attenuated. That means it is weakened, but it will be alive. So live weakened vaccine, or sometimes a killed vaccine, or substances from the pathogen, it will be inserted into the body. That type of vaccines, it induces the immune response, but it will not cause any disease. Some of the example first generation vaccines are measles, mumps, rubella, that's called MMR vaccine, and varicella vaccine, which is given for chickenpox. Second generation vaccines, we have the pure surface antigen of the pathogen. They are not going to insert the pathogen there. They will take only the antigen. The antigen, it will be on the surface of the pathogen. Those antigen alone, it will be given as a vaccine. That is called second generation vaccine. Whereas in the first generation vaccine, they will give attenuated form or inactivation. Killed pathogen, it will be given, or toxins. The toxins, after chemical modification, the toxins are poisonous substances produced by the pathogen, that it will be given to a person to induce immune response. The first generation vaccines means live attenuated, killed vaccine and toxoids. These are all examples for that. For killed vaccine, we have Salk's polio vaccine, the poliomyelitis virus, and DPT, diphtheria pertussis tetanus virus, tetanus. This is a vaccine which is given. They are all toxins, toxoids. That means the toxins after chemical modifications. Whereas third generation vaccines, it is the purest and highly potency vaccines. Here the vaccine is DNA vaccine or recombinant vaccines. These recombinant vaccines, it will be introduced into the body. The body itself, the vaccine itself, it prepares a body for a particular disease. They are all DNA vaccines. They will incorporated with our DNA and it induces the immune response. That's why they are called as third generation vaccines. Next, we are going to talk about hypersensitivity or immune response. Immune response. That's what I am going to explain to you after that. Okay. So now we'll go for a break. So during this break time, I want you to take a break. Just relax yourself for 5 minutes, and after 5 minutes, I'm going to explain to you about the hypersensitivity or immune response. Mahesh, do you have any doubt, Mahesh, till now? If you have doubt, please post your doubts. Welcome back, children. The next topic, what we are going to learn today, is about hypersensitivity or immune response. What is this hypersensitivity? Sometimes for some particles or some allergens, our body will start reacting to that. That is called as hypersensitivity. You suppose when there's a dust particle is there, immediately you will start sneezing. That it is also a response by the immune systems. So the exaggerated response of the immune system to certain antigens, we call it as allergy. Allergy means altered reaction. Altered reaction. So our body starts showing reaction to that. The substance that provoke the immune response are known as allergens. The allergic reaction, sometimes when you come in contact with some insect bite, the body, you can see that here and there you can have some patches like this. Or the food that what you have taken, if it is not good, it contains some poisoning substances, the skin it will develop rashes like this. So how long this response it will be there? Suppose you are exposed to dust particles, the reaction will be for few minutes. Sometimes few seconds. Whereas some reaction, it lasts about an hour. Sometimes for 2 to 3 days. The common allergens are the mites in dust particles, pollen grains, insect venoms, hay fever and asthma, sneezing. Some of the allergic responses that are shown by a body, sneezing it will be there. Sneezing, watery eyes. Sometimes when you take too hot foods or too spicy foods, the watery eyes it will be there. That is an allergic response. Running nose, difficulty in breathing in the time of hay fever and asthma, these are all allergic responses. So the allergic responses, it is a form of immune response that are mediated by immunoglobulin epsilon and mast cells. So these mast cells in a body and also the immunoglobulin epsilon, these are the antibodies which bring about the immune response. Usually what will happen, if there is any allergens are there in the body, your body start producing a substance called histamine and serotonin, which brings about the responses. Next, I am going to use a term called anaphylaxis. What is anaphylaxis? A sudden, systematic, severe, immediate response shown by a body, that is called anaphylaxis. Immediate, very quick response shown by the body. That's called hypersensitivity reactions. Sudden, systemic, severe, immediate hypersensitive reactions, which are mainly done by the mast cells. That is called anaphylaxis. Next, we are going to talk about the immune diseases. So far, we have learnt about the hypersensitivity or immune responses. Next, we are going to talk about the immune deficiency diseases. The immune deficiency diseases. So, what are these immune deficiency diseases? The diseases that is acquired a body due to lack of immune response or due to malfunctioning of immune system or due to deficiency of immune system, that is called immunodeficiency diseases. First, I am going to explain to you about the AIDS, acquired immunodeficiency syndrome. This is a disease that is mainly affects the immune system. The term itself, it is acquired, will get that will causes immunodeficiency and syndrome. What is syndrome? Collection of symptoms are called as syndrome. For example, if you have cold, sneezing, running nose, throat infection, these are all common symptoms of cold. Like that, lot of symptoms it will be there. Those collection of symptoms are called as syndromes. Collection of symptoms are called as syndromes. These diseases, the acquired immunodeficient diseases, it invades our body in a form of virus. That virus is called HIV virus. It's called human immunodeficiency virus. This virus, it enters into a body through sexual means or through blood transfusion. It enters inside a body, the blood transfusion from infected person, not from normal person. It enters inside the body and it mainly attacks the CD4 cells that are responsible for production of antibodies. The helper T cells are CD4 cells. These are all the cells that play an important role in production of the antibody. When this HIV enters inside the body, its main target is CD4 cells. It attacks the CD4 cells, which stimulates the antibody production by B cells. So there is no antibody production. When there is no antibody production, all the opportunistic infections due to failure of immune system, it will exaggerate. So natural defense against the viral infection, natural defense against the other diseases, it will be lost. The viral antigens, HIV, there are two types of HIVs, there type 1 and type 2. Both, it brings about immunodeficiency. This discovery of AIDS was first identified in homosexual individuals in the United States of America during the year 1981. This is actually a communicable diseases which can mainly spread from infected person to a healthy person through sexual contact, through blood and blood products, from infected mother to the child. These are the three possible routes. In the year 1983, Luc Montagnier at Paris Institute, National Institute of Health, USA, isolated the virus that causes AIDS. So AIDS virus was first isolated in the year 1983. In 1986, a committee of taxonomists of virus, they have coined the term immunodeficiency virus. This is a virus which belongs to the family called lentivirinae subfamily of retroviruses. What are retroviruses? These viruses, they'll have a single stranded RNA molecule, and along with that they have an enzyme called reverse transcriptase enzyme. These enzyme, they can bring that two single stranded RNA into a double stranded DNA forms. So retrovirus is actually RNA virus. Once it enters inside a body, our body we have the DNA virus, right? When this enters inside, the HIV enters inside the body, the HIV contains the RNA virus, retrovirus, single stranded RNA virus. Along with that, it has an enzyme called reverse transcriptase. Once it enters inside the body, it combines with that, it combines with our genome, and where the two single stranded RNA which is there in HIV, it is joined together and become DNA, and then it incorporated with our body genome. So the RNA of HIV, it will become DNA inside the body. The HIV is a spherical virus. It will be a 10 to 100 to 120 nanometer in diameter, and they have a lipid bilayer, and also they have the matrix protein, it will be there, which is a capsid, and inside you can see that there will be a capsid protein, and then the nucleic material, it has two single stranded RNA along with reverse transcriptase enzymes. And also you can see that the GB41 and GB120, these are all the glycoprotein spikes that are present in the outer region of the HIV virus. Glycoprotein 41, glycoprotein 120, and then they'll have a core capsid protein, it will be there, and also the outer they'll have the matrix proteins. They are well protected structures. Now I am going to explain to you about the HIV transmission, how HIV is transmitted from one person to another person. HIV is often, we can see that it is located within the cells, mainly in the macrophages, and HIV can survive within our body, within the host cells, for about 1.5 days inside the cell, but outside the body it will be alive only for 6 hours. Once it enters inside a living cell, it can live for about 1.5 days, whereas outside environment it can be alive only about 6 hours time. What are the various means it can be transmitted? It is transmitted through unsafe sexual contact with infected person, blood transfusion, during that time contaminated blood transfusion or contaminated needles or contaminated surgeries, organ transplantation from infected person, blood transfusion, and also the vertical transmission of blood from mother to the child, infected mother to the child. That's called fetal route. These are all the various means of transmissions. So once this virus enters inside the body, as I mentioned that there are the virus, it has the nucleic material, nucleic acid as RNA. This RNA, once it enters inside our cell, because of the reverse transcriptase enzyme, it will become DNA. This DNA, it incorporated with our body genome. After that, it start producing lot of RNAs. Our body itself start producing lot of RNA genomes. These RNA genomes, it comes out of the body and start infecting the other cells. So this is how the viral infection, it is increased in the body. The viral DNA, it enters inside, it become RNA become DNA, the DNA incorporated into the host cells and infect the cell to produce lot of matured HIV viruses or viral particles, which further start infecting the other cells. These macrophages continuously produce viruses, because as I mentioned that the macrophages only you can see lot of the HIV viruses. The HIV enters into the T lymphocytes. Inside the lymphocyte, it start CD4 lymphocytes, it start producing lot of multiple copies of that, and it can come out and invades the other cells. When it started infecting all the CD4 cells that are involved in production of the antibodies, it will be deficient. That's why it is called human immunodeficiency virus. Due to this, what will happen? The lot of symptoms as well as the opportunistic infections can be there in humans. During the period of infection, fever, it will be there, diarrhea and weight loss, drastic weight loss, it will be there. Due to decrease in the T lymphocytes, because the HIV, it attacks and kills the CD4 cells, the person start suffering from other opportunistic infections like tuberculosis, candidiasis, salmonellosis, tuberculosis, pneumonia and other bacterial viral diseases. These are all opportunistic infections. Test for HIV. ELISA, enzyme linked immunosorbent assay, it detects the presence of HIV bodies. It is a preliminary test. But confirmatory test is called Western blot test. Western blot test is more reliable and is a confirmatory test because that is the one which detects the viral core proteins. You know that the RNA is surrounded by the core proteins, like 2 proteins it will be there. It can able to identify that. If both the test, the presence of antibodies are there, the presence of antigens, it is there, the person is considered as HIV positive. That means the person is infected with HIV. Prevention of AIDS. There are a lot of organizations throughout worldwide, they are involving in creating awareness and to prevent the AIDS. We have in our country National AIDS Control Organization along with World Health Organizations. They are conducting a lot of awareness programs because AIDS is not cure but it can be prevented. So prevention is better than cure. So they are creating awareness about having a safe sex, promoting regular checkup and safe blood transfusion, safe surgeries, proper disposal of the infected needles and uses of condoms during sexual contact with infected persons, and prevention of drug abuse because the drug they are using the same needle, during that time the transfusion is more. The AIDS awareness program which was conducted by National AIDS Control Society, and also in Tamil Nadu we have Tamil Nadu State AIDS Control Society organization and other NGOs along with the World Health Organization. They are creating a lot of awareness about prevention of the AIDS. That is about AIDS. Next we are going to talk about autoimmune diseases. Children, do you have any doubt in AIDS? Prevention of AIDS? Immunodeficient disease, do you have any doubt, children? Okay, after 5 minutes I am going to explain the autoimmune diseases. Autoimmune diseases and then tumor immunology. I am going to explain about tumors.